So just to be clear, Tuesday you have a test, and it has to be Tuesday, so it's not like I'm going to be able to change it. All right, so as I say, Tuesday, there's no way around it. Tuesday's the latest day I can do it. You have an exam on dihybrid crosses. And the chapter reading. I'm going to go over it today. I'm going to give you the answers that you should have had on your practice problems that you've been working on theoretically for the last 3 days, that you should have been working on in small groups, that you should have read your chapter on and turned in already. All that should have been done. So let's go through the answers, and then we'll go through the chapter reading as well, and then we're done. And you'll know what's going to be on that test on Tuesday. Yes, I am. I'm posting it, but also remember your chapter has practice problems, so take a look at those practice problems in the reading. So it says here, if we read it, you have a Punnett Square, you have a cross between this, if you cross a heterozygous male for tallness, the homozygous recessive for female for tallness, then give the genotype for and phenotype or the ratios, phenotypic ratios. So heterozygous male, which means if we're talking about tall, it means it's big T, little t. And it says homozygous recessive for the female, so that's little t, little t. You might ask, well, how do you know it goes on top? How do you know it goes on the bottom? It really doesn't matter since the math works out either way. You end up with the result of big T, little t, little t, little t, big T, little t, and little t, little t. In this cross, you end up with 50% short, little t, and 50% homozygous tall, or heterozygous tall, no homozygous tall, right? So your genotypic ratios is 50% little t, 50% big T, little t. Your phenotypic ratio, again, those vocabulary words always come back to haunt you. Your phenotypic ratio, and that's what this means here, phenotype ratio, is 50% short and 50% tall. All right, so let's go ahead and look at this. Now remember, if you haven't turned it in, you can't turn it in now. It was due at the beginning of class. So just to be clear, if you're trying to copy these so you can submit it at the end of the period, that's not going to work. So this is just us going over the answers.
Wait.
All right. In garden peas, big T is tall and little t is short, and big A is auxiliary flowers, and little a is terminal. What are the expected genotypes and phenotypes of the offspring if a heterozygous tall, and what would that look like? Well, they tell you it would look like this, right? Heterozygous tall, what is it? Heterozygous auxiliary plant is crossed with heterozygous tall, and having a terminal plant. Now they tell you terminal, and you're like, well, how do you know if it's homozygous or heterozygous? Some of the questions that kids have asked me, those students have actually been working over the last 3 days. They get questions like, I get questions like, okay, how do you know how do you know what the terminal plant is? It heterozygous or homozygous? Well, you know it has to be homozygous. Why? Does anybody know? Why must the terminal allele be homozygous? Because it has to be two little a's. You cannot get terminal unless it's a recessive trait. So all recessive traits have to be homozygous. So that's what you're crossing. They don't tell you if it's male or female because it really doesn't matter for this problem, but there it is. Now, of course, you have to figure out what the gametes are, right? What do the gametes look like? so far what we talked about is these are the parents, these are the diploid cells, what they look like. Now we have to figure out what the haploid cells look like. What do those sex cells look like? Well, for the male, it's going to be big T, big A. And you can do FOIL. I'm just going to do it off the top of my head. You're going to have big T, little a. You're going to have little t, big A. And you're going to have little t, little a. So all four possible gamete combinations are possible because the male is heterozygous for both genes. The female, on the other hand, the female is going to have, well, it could have big T and little a, or it could have little t and little a, because that's all it has. It only has little a's. There's no big A involved for the female. Note that the female has no big A, just two little a's. So there's only two options. I don't need to worry about these down here. This and this can be ignored because there's only two kinds of eggs that female can make. Does that make sense to everybody? Do I have any questions? Is there any concerns at this point that you don't understand what's going on? Yeah.
So if you keep going and do two big T's with two little A's and two little T's with two little A's, you can't do that? So that's wrong?
No, you can do that.
Wait.
You can do, so she asked the question, what if you put big T, little a, little t, little a here, and just have all four, and yeah, you can do it, you're just repeating your work, right? Your numbers are all gonna work out the same, so it doesn't matter, you'd be right, you're not wrong, you're just wasting some time. So when you're on an SAT or ACT or a college exam or AP biology exam or even an exam here at the end of the year, your end of course exam, time might be an issue for you. If you feel more comfortable doing it all and just makes you feel better so you're not making mistakes, go ahead and do that. It works anyways, yeah.
I don't know where to turn these in at, like on which one.
All right, it says 3-day dihybrid problem practice.
For this one?
For the practice problems. And then the notes on the chapter say notes on the chapter.
Also, both of these go on the
On one, both go on the practice problems, that's correct. Okay, so now, when we're doing the, just making sure that the dates represent what they're supposed to. When we're doing then the actual Punnett Square, we're trying to figure out what we got a big T from dad, and we got a big T from mom, and then we got a big A from dad, and a little a from mom. That's what we got. Notice I put the T's together and the A's together. That makes it easier for me to read. And I'm gonna stop with the color coding now because it's gonna take more time than I really would like. So big T, big A, little a, big T, little a, big T, little t, big A, little a, and big T, little a, little a. Notice there's no homozygous big A because you have because mom was homozygous recessive. Big T, big A, little a, big T, little a, little a, little t, big A, little a, little t, little a, little a. So then when we're talking about the percent, when we talk about offspring and genotype percentages, well, how many do we have in total? 8. So some of you might have had 16, but as you do the fraction, you'll see the fractions will be the same. All right, you might have a bigger number, but when you reduce it, they're all the same. So we have big T, big A, little a. So when I look at this and I look for big T, big A, little a, that's 1. And that's the only one I have. So what would that be? That'd be 1 out of what?
1 out of 8.
1 out of 8. Now, some people might have done all 16, so they'd get 2 out of 16. But 2 out of 16 reduced is 1 out of 8. So it's the same result. So when we take a look at this one, big T, big A, little a. That's how many? We got this one. Are there any others? There is, over here. Do you see it? So there's 2. In fact, if you'd like, I'll do a different color highlight. There's 2. So that's 2 out of 8. And that'll reduce to 1 over 4, if you like. And if you do the same, if you did it with 16, it'd be 4 out of 16, which would reduce to 2 out of 8, which would reduce to 1 out of 4. So you'd get the same. So then the next one is big T, little a. There's only one of those. Do you agree? Big T, little a. There's only 1 out of 8 there. And then we have big T, little a. There's 2 of those. So that's big T, little a. There's 2 out of 8 there, which would be equal 1 out of 4. And then you have big little t, big A, little a is 1 out of 8. Is that correct? Big T, little t, big A, little a is 1 out of 8. And the last one's little a, little a, which is 1 out of 8. Does everybody see that? Does anybody have a problem seeing that? Remember, you have to do this on a test, so this is the time to say, I don't get it. I don't see it. What's going on? All right. Seeing as that no one has an issue. Now, the phenotype, what kinds of phenotypes do I have here? Well, let's look at it. I got this one here is tall with axillary flowers. So this is tall with axillary flowers. This one's tall, even though it's little t, has a big T, so it's tall with axillary flowers, tall with axillary flowers. All right, so that's 3 out of 8. 3 out of 8 are going to be axillary flower and tall. I'm just going to put A just so that I can write without too much trouble. Everybody okay with that so far? Why did I say 3 out of 8? Does everybody see the 3 out of 8?
Yes, because it's 6 out of 16.
So you got, look, tall and axillary, the big T and the big A, tall and axillary. Tall and axillary. Do you see any others that are tall and axillary? Axillary means it has a big A, tall means it has a big T. Are there any others on there that have a big T and a big A? No. So it's 3 out of 8 are tall and X. I'm just going to put X. Is that okay? Mm-hmm. All right. How many of these are tall with terminal flowers? So let's count them. This is big T, so it's tall, as a, little a, so it's axillary, terminal. Here's tall and terminal. Here's tall and terminal. How many do we have? 3 out of 8. Again, they're tall and axial. Finally, we got how many are tall? Well, there's only going to be 1 out of 8. There's only 2 left. So 1 out of 8 is going to be short and axial, right? And the other is going to be short. Oh, and I put, dang it, guys, I'm sorry. I put axial here. Nobody caught me. This should say terminal, shouldn't it? You don't know? Terminal is little a, little a. Axial is big A. So 3 out of 8 are axial, big A, and tall. 1, 2, 3. 3 of these are tall and terminal. Little a is terminal, so 1, 2, 3. 1 out of 8 is short and axial. Short because 2 little t's, axial because of the big A. And 1 out of 8 is going to be short and terminal. Why is it short and terminal? Because you have 2 little t's and 2 little a's. That's what should have been done on the first page of your work. Now let's move on. Here it says the horses of a coat color black is dominant over chestnut. Chestnut's a brown. The trotting gait is dominant over the pacing gait. That's just how they move. So big T is trotting and little t is pacing. If the homozygous black, what does that look like? That means big B. Let me zoom in on this so I have room. That means big B, right? Homozygous black. Pacer, so it's a pacer, which means it's little t. How do I know it's little t? Because if it's a pacer, it has to be little t, because that's the recessive trait. And up to now, we've only been discussing dominant recessive. Is mated with a homozygous chestnut. Now again, it's kind of repetitive because in order for it to be chestnut, it would have to be homozygous, little b. We're crossing these. And heterozygous trotter, which means a big T would give it trotter. How do you know it's not big T? Because they're telling you it's heterozygous, big T, little t. That's what heterozygous means. Heterozygous means, this word here means that this is big T, little t. Because what's the other option? It could have been waiting. It could have been, instead of big T, little t, it could have been what? If it's a trotter, what are the two possibilities? That's right, big T, or big T, little t. But how do you know it's big T, little t? Because it says what? Heterozygous. All right. What were the probabilities of genotype, phenotype of F1 generation? F1 means the first babies, the first cross. F2 would be the cross from the babies, babies. F3's the babies, etc. So how many different gametes can this one make? How many different gametes can this parent make? One. Big B, little t. That's all it can make. So that makes my life easy. I can just put big T, big B, little t. Then I just have 4. Now, some of you, I'm sure, did all 4 on the side and all 4 across. That's fine. But again, look how easy it is when you don't do that. How many different babies can this one make? 2. Little b, big T, or it can make little b, little t. So that means it's only 2. Little b, big T, little b, little t. So either it's black, heterozygous black with heterozygous trotting, or it's heterozygous black with a pacer. A pacer's heterozygous black. Those are the only 2 options. 50% are this, and 50% are that. That's it. We're done. Genotype, that's the genotypic ratios. It's also the ratios for the phenotype. So the phenotype, the genotypic ratios, big B, little b, big T, little t, and the other one's big B, little t, 50% each. The other one is it black and a trotter, or is it black and a pacer? This is black and a pacer, this is black and a trotter, 50% each. Okay. F1 offspring, genotype, percents, and ratios. I just did it, so there's nothing else to discuss. You can do 1 out of 2 and 1 out of 2, and then that equals 50%. I mean, whatever you want to do. I did it all in one step, but does everybody get that? I don't need to do the rest, right? I don't need to write it out. Let's go on. All right. In rabbits, the coat color big B is dominant, brown, little b, short hair, big S, long hair, little s. In a cross between homozygous black, short, homozygous black means what? Big B. Short is dominant, homozygous short means is big S, is crossed with, and that's a male, that may or may not be important. If it's X-linked, it's important. Is crossed with a brown, and a brown would be little b, and long-haired female, which would be little s. How many different gametes can the father make? 1, big B, big S. How many different gametes can the mother make? 1, little b, little s. So the cross is real easy. It's only one possibility, big B, big S, little b, little s. Big B, little b, big S, little s. All 100% of the babies are going to be black with short hair, right? 100% of the babies are heterozygous for both genes. So it's 100% the ratio is 1 out of 1, 100%. That's it. There's no other discussion. Real simple. Again, you could have done all kinds of things to get the same answer, but that's the easiest way of doing it. Yeah. Yes. Imagine a couple is planning to have children, a male heterozygous tongue roller and homozygous dominant unattached earlobes. So now they give you the letters. The tongue roller is big R, non-roller is little r, unattached is big U, and attached is little u. So they say heterozygous tongue roller, so that's big R, little r. And it says homozygous dominant unattached is big U. So it's homozygous, so it's big U, is crossed with a female that's homozygous recessive for tongue rolling, so that's little r, and heterozygous for unattached, big U, little u. Not us, but little u. How many different gametes can the first parent make? This parent. 2, correct. Big R, big U, and what? Or little r, big U. And how many will this one be able to make? 2, little r, big U, or little r, little u. That's it. So now, do your Punnett Square. You got big R, big U, little r, little u, little r, big U, or little r, little u. Is it time to go?
Yeah.
All right. So we got big R, little r, big U, little r, big U, big R, little r, big U, little r, big U, little u. And so now we have a little bit of deeper discussion, a little richer discussion, because there's more options. So what will this one be? What will this one be? A tongue roller and what? Attached earlobes. What about this one? A non-tongue roller with attached earlobes. What about this one? Tongue roller with attached. So this is actually going to be the same phenotype as that one, and this one will be the same phenotype as that one. So these two actually have the same phenotype, even though their genotypes are different. That's really key. All right. So the Austrian Genotype percentage and ratio, you have 1 out of 4 are going to be big R, little r, big U. 1 out of 4 are big R, little r, big U, little u, heterozygous for both. 1 out of 4 is little r, big U, and 1 out of 4 is little r, big U, little u. And then, of course, the phenotypic ratio is easy. It's 50% or 1 out of 2. Roller, and attached, and 50% non-roller and attached. That's it. That's all we have time for. I will finish this and post it if you need help for study purposes. You know you've already submitted your answers. Good luck. So we got this problem set's pretty easy because they give you this. When you have a word problem, I would suggest strongly that you do this. You know, big G is this, big G little g is this. Anytime you have a word problem that's confusing, if you do this, it makes your life easier. It makes sure you don't make a mistake with what's what. What are the phenotypes of the rabbits that described here? The following genotypes, right? So how would you describe, this? You would describe it with gray hair and having black eyes.
It's red.
Oh, thank you. Red eyes. And then the other one's white hair. And the other one's black eyes. And white hair. And red eyes. And you see, this is pretty straightforward. There really isn't a lot to it. You just have to know how to read it. As long as you put your effort into it, it shouldn't be an issue. Effort being you actually trying to do it. So the male rabbit, then this cross, they even did this for you. I'm not going to do it all because I don't know what else to do. It's big G, big B, little b, etc., right? And I don't like doing it this way because you're repeating. Yeah, you only need, like, this whole thing here, I wouldn't even do it. And I wouldn't do this thing here either, because they're just repetitive. I know they give you the spaces, but I just wouldn't do it. Big G, little b, big G, little b, little b. And this is all, of course, all going to be the same. Yeah. Because honestly, let me redo that. Because I think it's worth us discussing this. Because we just did it on the previous page, but honestly, not for nothing, you learned something. So I would not do this. I would not do this. I wouldn't do any of these.
So it's just two.
So all you really need to do is 2, because there's only 2 possible outcomes. Come on, people. So how many out of 16 have gray fur and black eyes? Gray fur and black eyes. Well, it would be 50%, or 8 out of 16, right? How many 16 out of 16 with gray fur and red eyes? 8 out of 16. How many? 0 and 0.
No.
All right. Have a good day, guys. A great weekend. Make sure. Now here again, let's take a look at. Let's take a look at what they're asking here. They say a male rabbit with genotype big G, big B, little b is crossed with a female rabbit that's big G, little g, big B, little b. Now, automatically, if I did my Punnett Squares and I did my homework and I listened in class and I took notes and I reflected, then I know that I have a heterozygous for both traits here and a heterozygous for both traits here. So I know I'm going to have 4 different gametes. So I'm going to need all 16 of them. So I'm not going to get the opportunity to knock off repeats because there's going to be 4 different gametes for each parent. So that means the first one is going to be the second one. Let me just pause here and just do it. Now that you see that we've done all the genotypes, now they're asking you, what are they asking you over here? What's this about? Are these genotypes they're asking you? What are they asking you over here? the phenotypes. So you have to ask yourself, how many of these, they ask, out of 16, and there's 16 here, how many of these have gray fur and black eyes? That's a big G and a big B. Any big G and a big B. Gray fur with black eyes. That's this one.
No.
That's one. There's two. And you know that's double. And I could go ahead and count, right? There's two ways I can do it. I'm going to do it both ways, actually, now that you said that. The easy way, if you remember what I told you, was when you have two heterozygotes mating, you're going to have a 9:3:3:1 ratio. Like if you remember that ratio, then you know the answers right away. 9 that are going to be double dominant phenotype, 3 that are going to be dominant on one phenotype and recessive on the other, 3 that are reversed the first set, and 1 that's going to be recessive on both of the phenotypes. So let's go ahead and count them and see if we're right. So here's the dominant 1, 2, and 3, and 4, 5, and 6, and 7, and 8, and 9. 9 are dominant for both traits. The next is how many have gray fur with red eyes. It's dominant for gray fur, but recessive for red eyes. That would mean little b, with any big G. Well, here's 1, here's 2, and here's 3. Those are the only 3, and that matches. Then the next one is how many have white fur and black eyes. That would mean little g for white fur, and a big B for black eyes, because black is dominant and the white fur is recessive. So that's 1, 2, and 3, and you see there's your second 3. And lastly, there's only one that's both white and blue and red eyes, and that's that little g, little b, and that's this one. So you see the phenotypes match this 9:3:3:1. If you remember the ratio, the one ratio I told you was going to be very important, it pretty much always works out that way, because mathematically it's the only way it can work out. All right. Is there any questions as to this? You will see this on the test tomorrow. So please let me know now if you have a question. You've had this now for a week. You've gone over. You did it in class for 3 days, along with taking your notes on your chapter. So really, you've had every opportunity in the world to work out any kinks. So show the cross between little g, big B, little b, and big G, little b, little b. Again, how many different kinds of gametes can this parent make? 2. Little g, big B, and little g, little b. And this parent over here can make how many? 2. That's big G, big B, and big G, little b. So this parent can make 1, 2. That's all it can make. And you can do FOIL, and you can figure that out. And this other parent can make this. And that's all there is there. As simple as can be. Big G, little. Of course, because these are both big G and big B, all the sperm or eggs, whatever this is, all the gametes that use this genotype are going to be dominant in their phenotype. So you have this going on, and this. You have this going on. And of course here, dominant for both phenotypes here. However, they're going to have one individual that's going to have the recessive phenotype for red eyes. Other than that, they all have the dominant phenotype. All right. So you should have been able to do that pretty simply. An aquatic arthropod, which is an insect, right? With an arthropod is an organism that has an exoskeleton. It could be an arachnid or it could be an insect. I'm glad you guys are going to continue to have this conversation while I'm explaining. The allele barbs, big B, is dominant over smooth, and the organism is not resistant to pesticide is a big N. So if it's not resistant to pesticide, that's normally how these organisms come. It's dominant over the recessive that are resistant to pesticides. And the dominant allele for barbs is dominant over the smooth. The rough is dominant over the smooth. So if you have this genotype, what phenotype would you have? Barbs. And I think you get the rest of this. I don't think I need to. If you don't know this, if you have a problem with this, you really need to see someone, myself or someone else. You should have already gone and seen someone at this point, because this is not. And the last is it's resistant. All right. I think that seemed fairly straightforward, yes?
Very straightforward.
I don't know what else to say. Cyclops is that is resistant to pesticides. It has smooth antennae. Is crossed with one that is heterozygous for both traits. Smooth, remember, is recessive, right? So that would be little b. And heterozygous means what? That it's going to be barbed. Yeah, they're different, that's right. But because they're heterozygous, they have the dominant phenotype. And it says that is resistant. Cyclops is resistant to pesticide as smooth. So resistance means what? It's little n. It's cross one that heterozygous for both traits, so it's not resistant. Big N, little n, and then also big B, little b. So you have big B, little big N, little n, cross with little b, little n. So set up the Punnett Square. Well, you need to know the gametes. So the gametes for the heterozygote, you'd use FOIL, and you'd end up finding it's big B, big N, big B, little n, little b, big N, and little b, little n. And of course, this parent can only make one type of gamete, thank you, little b, little n. So you end up with this going on. What are the phenotypic ratios of the offspring? Well, are there any phenotypically? Let's take a look at it. This one's barbed and it's not resistant. This one is barbed but resistant. This one is smooth but not resistant. And this one is smooth and resistant. So it's 1/4 each. 1/4 barbed and non-resistant. 1/4. By the way, anyone that turns this in after I finish with all this, I'm taking points off, right? Because I'm giving you all the answers, and the kids that already submitted their work without someone giving them all the answers, I'm sure they did it on their own. Does that make sense? 1/4 barbed and resistant, right? And 1/4 smooth non-resistant, and 1/4 smooth resistant. Yeah. Yeah. So that seems simple to me. Do I really need to go through all these? Do you want me to go on through the chapter? How would you like us to use the time we have left? The chapter, you're going to have questions on the chapter reading as well.
Yeah, let's go through that.
Because this just seems to me like it's repeating the same problem set, right? I think am I recording this? Yeah. For genotype, in this first problem, there's either this one, this a different genotype, this a different genotype, and this a different one. So there's actually 4 different genotypes, and if you're asked for a genotypic ratio, then you'd have 4 different ones. You'd have 1/4 for genotypic ratios. You'd have 1/4 big S, big W, little w. You'd have 1/4 big S, little w, little w. You'd have 1/4 little s, big W, little w. And you'd have 1/4 little s, little w, little w. That's where you have it. That's it for genotypic ratios. But then phenotypic ratios, how many different kinds of phenotypes do you have? You have the spotted skin with woolly hair, the spotted skin with woolly hair. Then you have a spotted skin with non-woolly hair. Then you have a non-spotted skin with woolly hair. And you have a non-spotted skin with non-woolly hair. So you have 4 different phenotypes as well. And you end up with 1/4 each of those as well. So 1/4, 1/4, 1/4, and 1/4. Does anybody have a problem with that? Because this is you're going to see a question like this on the test tomorrow. So when we look at, in horses, black is dependent on big B, chestnut, little b, trotting versus pacing. And they tell you that a black, homozygous black, which means what? Big B, pacer, and by pacer they automatically tell you that you have little t. Is that correct? Because in order to have the recessive phenotype, you could only have, it must be homozygous. It's crossed with a homozygous chestnut trotter. So that would mean little b, big T, because they tell you it's homozygous. Anybody have a problem with any of this so far? How many gametes can this parent make? 1, little b, big T. How many gametes can this parent make? 1, big B, little t. So you only have 1 cross to worry about. Big B, little t, little b, big T. All of their children will be big B, big T, little t. 100% genotype, 100% big B, big T, little t. 100% black trotter. Yeah. Yeah, no, I don't care if you're right. I just want to make sure we're going over every possible way of looking at this so that nobody is confused on a test tomorrow. So summer squash, white fruit color, big W is dominant over yellow fruit color, little w. The disc shape, big D, is dominant over sphere shape, little d. If a squash plant true breeding for white disc shape, what does true breeding mean again? That means they're homozygous. 100% of their children would, if you mate two of them, they would end up with the same phenotype because their true breeding means homozygous. Squash by true breeding for white disc shaped fruit is crossed with true breeding with yellow sphere shape. So that means that everybody's homozygous. So white W, big W, disc shape, which means big D, is crossed with yellow, little w, with little d. Is that true? Yeah. All right. So how many different gametes can each parent make? One. Big W, big D, and little w, little d. Again, big W, big D, little w, little d, easy peasy. 100%. And 100% what? White this shape. That's all there is to it. I mean, see, it's really turns out real simple as long as you know what kinds of gametes. This is why we did the dragon genetic stuff, so you understand what kinds of gametes is it possible for this creature to make. The whole point of this is to give you practice.
Back to what you just did.
All right, so here you have in mice, the ability to run normally is is a dominant trait. Mice, the trait is called running. Mice are recessive, causes mice to run in circles only. That's sad. Mice with a trait, they're called, we had little r, right? Waltzing mice, that's cute, but sad. Black-haired is dominant over brown hair, right? So little r, running in circles only. So for each of the following problems, determine the parent genotypes, determine possible gametes, then construct a Punnett Square to solve. All right. Cross between a heterozygous running, that means what? Two different, so big R, little r. Heterozygous black, which is big R, little r, then what? Big B, little b. With a homozygous running, which is what? Big R, and homozygous black, which would be what? Big B. So parental genotypes are big R, big B, little b, crossed with big R, big B. Possible gametes, well, it depends on which parent, right? So I'm going to move it over here so it's neater. So this parent number one, I don't know if it's male or female, can make, do your FOIL if you need to. Big R, big B, big R, little b, little r, big B, little r, little b. Those are four that this parent can make. And then the last, obviously, that this parent here can only make one, right? Big R, big B, that's it. So if you're going to go ahead and do that, you'd have to put big R, big B here, and then put your four across. Big R, big B, big R, little b, little r, big B, little r. Oh, I'm sorry, that's right. I was right. Big R, or little r, big, little b. So you combine them, you get this. Does anybody have a problem with any of this? Please speak now. Please ask your questions now. Write them down and pass them up if you're embarrassed. I don't care. You need to know what's going on here.
We have questions on the video. Can we ask them
I'd like to get on to the reading as soon as I finish these.
Okay.
All right. So what do we got? So for genotypic ratio, you got 1/4 of each. Do you agree? There's 4 different genotypes. Does everybody agree with that? Everybody see that? So you got 1/4 for genotype. So there's 2 things you have to think about. Genotypic ratio, and that ratio is 1/4 big R, big B, 1/4 big R, big B, little b, 1/4 big R, big B, and 1/4 big R, big B, little b. Phenotypic ratio, and this actually, we just keep going over and over the same thing. Does everybody agree that this is all very repetitive? It's just different examples of the same material. Once you learn how to do it once, you can apply it to all of them. It's really easy. So again, R is running, little r would be waltzing, big B is black, and little b would be brown. So when we look at these, this is running black, this is running black, this is running black, and this is running black. So even though you have the genotype is 1/4 each, right? Your phenotype is 100% running black. Pretty straightforward, correct? All right. Yeah. You could write like 100% running black, or all are running black, or you're going to get a multiple choice test tomorrow, so you don't have to worry about like stating it. But it all. If somebody says, if they ask you a problem on the state exam and they say how many of the 16 would be, how many out of 132 would be each different phenotype, then you say 116 would be running black and 0 the rest, right? Let's be clear. The state will expect you to type in your answer. Not all your answers are multiple choice on the state exam. Are we clear? Across a homozygous black mouse with a homozygous running, so it means what? Big R, homozygous black, big B. With heterozygous running, big R, little r, with brown mouse, which is what? Little b. How many different so the parental genotypes are big R, big B, and it's being crossed with big R, little b. The possible genotypes for this one is only 1, so this parent can only make 1. Do you agree? Big R and big B, that's it. And this parent can only make how many? 2, big R, little b, or little r, little b. So this, and then you'd have this and this. So you'd have possible zygotes of big R, big B, little b, big R, big B, little b. So 50% of each as far as genotypic ratios go. You'd have 1 out of 2 would be big R, big B, little b, and 1 out of 2 would be big R, big B, little b. But phenotypic ratios, as before, would be what? What would it be? 100% running black. Why? Because there's no way that a parent that can only make the genotype of a homozygous dominant is only going to give its dominant genes. So all its children are going to be dominant. A cross of a waltzing mouse, so now they use the word waltzing. Now I hope you know that means little r. with a waltzing what? They don't tell you. Oh, brown. Brown, which is little b, waltzing, with a waltzing. So there's only one, so the parents are this. There's only one possible gamete. So there's only one thing that these things are going to be able to make. They're waltzing 100%, little r, little b, and 100% waltzing brown. Does everybody agree? You know, I make mistakes doing this, so if you see something you disagree with this, it's a good time to ask. So I think these are, it seems like I looked at the ones that were turned in, and if you need for you to check, people seem to know what they were doing. No one has really any kind of difficult questions. They're fairly straightforward. Cross homozygous running with heterozygous black. You do have tutors available twice this week. You do have me before school, after school. If you have any problems, of course, we're running out of time. Homozygous running would be big R. Heterozygous black would be big B, little b, and waltzing brown would be little r, little b. So the parental genotypes are big R, big B, little b, crossed with little r, little b. The possible genotypes for this parent are what? 2, which are big R, big B, or big R, little b. The possible genotypes for this parent are only 1. So when we do the Punnett Square, we would get 1 little r, little b. Could be make up to 2 different ones. But there are 2 phenotypes this time, so 50% and 50%. So 50% little r, big B, little b, and 50% little r, little b. So that would be 50% running black and 50% running brown. Are we good with that? And next, cross heterozygous running brown mouse. Heterozygous running would be big R, brown, little b. Heterozygous running, big R, little r. Homozygous black, so it's big B. So the parental R, big R, big B, crossed with big R, big B. The possible gametes for this parent are what? Big R, little b, or little r, little b. The possible gametes for this parent are what? Big R, big B, little r, big B. So you have 2 different ones that are possible here. So there's 4 possible zygotes. Am I losing anybody? Does anybody feel like we need to go over? Am I going too fast? Give me some feedback, people.
It's just repetitive.
Thank you. That's how I feel too. But I have to say, I just want to make sure that there's a reason you have all these repeated examples, so you practice, right? No, I know why you're saying it. I just want to make sure everybody knows that you're expected to practice these. So, of course, you got 1/4 each of these genotypes, right? Mm-hmm. No, you don't. What do you got? You have 50% this genotype. Then you have 1/4 this genotype and 1/4 this genotype. Do you see that? Yes. All right. I'm not going to write it out. I've written it out already before. So you have 25, 25, and then 50. What about phenotypes? So you're going to have 3/4 are going to be with the phenotype. 3/4 of them are going to be running and black, correct? And one of them, 1/4, is going to be waltzing and black. Do we agree? All right, pretty straightforward. Cross with a heterozygous running and heterozygous black mouse with a heterozygous running. So that's big R, little r, big B, little b, cross with it, big R, little r, big B, little b. Now you got a full on heterozygous for both traits, which would mean, I'm not going to write them in the blanks. I think you can handle that. It'd be big R, big B, big R, little r, big B, little r, little b, all the way down, right? I almost don't want to do this because I feel like you all know this, and we could be working on material that's a little harder, or at least a little newer. You get this, yeah? I'm not going to finish this last one. On this problem set here, it says there's a Punnett Square using the following information. They tell you in the problem set that tall plants are big D, dwarf plants are little d, purple flowers are big W, and white flowers are little w. A cross between homozygous dominant parents, and they even give you the genotype, is crossed with a homozygous recessive, and they give you the genotype there. Well, how many different kinds of gametes can this parent make? I hope everyone at this point says just one, and that's big D, big W. And this parent can only make little d, little w. So really all you have is big D, big W crossed with little d, little w. So what you get is big D, little d, and big W, little w. That's all 100%. So they ask you, using the Punnett Square for question number 1, what is the probability of producing tall plants with purple flowers? It's 100%. Tall plants with purple flowers, tall and purple. What are the possible genotypes? Only one genotype is possible for these plants, and that's this one. What's the probability of producing dwarf plants with white flowers? 0 and 0. There's nothing that could have white. There's no way to get white plants, white flowers, and dwarf plants with this genotype. What's the probability of producing a tall plant with white flowers? Again, 0. You can't have a tall plant with white flowers with these parents, and that's not applicable. NA. Probability of producing dwarf plants with purple flowers, 0%, NA. It's just a matter of, do you understand that this parent can only produce this gamete, and that this parent here can only produce this gamete? So if you understand that, then I think you understand that percentage and where it comes from. It says set up a Punnett Square using the following information. Again, it goes through and gives you the big B is for this. It's a black, and little b would be white for big R is rough, and little r is smooth. So this is what? This is a black, rough. For phenotype, and this is going to be a black rough. So if you set up the phenotype, the Punnett Square, you would go ahead and do your gametes. Which gametes could the big B, little b, big R, little r make? That would be big B, big R, big B, little r, little b, big R, and little b, little r. And you can do that across the board, up and down. And of course, you would end up with this. Let me go ahead and pause it here. So now that we have the Punnett Square filled out, they're going to start asking you questions like, what's the probability of producing a guinea pigs with black and rough fur? Now that's what? A phenotype that has both dominant phenotypes. Is that correct? Yeah. Now, if you remember correctly, I told you with dihybrid crosses, and as you watch this video at home, if you go home to watch it to prepare for the test, I told you probably 2 weeks ago when we first started this, when we do this dihybrid cross and you're looking at phenotypes, that if both parents are heterozygous for both genes, then you're going to get 9 that are phenotypes that are dominant phenotypes for both traits. You're going to have 3 that are dominant for one phenotype and recessive for the other. 3 that are reversed, they're recessive for the first phenotype and dominant for the other. And only 1 that's recessive for both. So how many of these would be black and rough? That's a big B and a big R. Just a single big B and a single big R is all you need.
9.
So this is 1. And we know it's 9. We know it should be 9. Let's count. 1, 2, 3, 4, 5, 6, 7, 8, 9. And it's the same every time. It's 9. How many of these are black fur but smooth fur? Black and smooth fur. So that means big B and little r, little r. There's a big B with little r, little r. There's a big B with little r, little r. And there's a big B with little r, little r. So that's 3. How many of these are rough? White with rough. In other words, white being little b, and rough being a big R. There's 1, there's 2, and there's 3. So 3. How many of these are white and smooth? Only 1. Do you see that? Yes or no? All right. So when you're looking at these, you're looking at that 9-to-3, 3-to-1 ratio becomes very useful on a multiple choice exam, on a state, on the SAT, ACTs. Anytime you get any kind of question about a dihybrid cross that asks you a simple, hey, this is they're heterozygous for both genes, they're being crossed, you can do the whole Punnett Square if you want, or you can just remember 9-to-3 to 3-to-1. Be very careful. Make sure you're dealing with this kind of problem. There could be all kinds of little tricks in these word problems, correct? All right. So, possible genotypes, we know what the possible genotypes are. They're right there. I'm not going to go ahead and rewrite them. What is the probability of producing guinea pigs with black and smooth fur? 9 out of what? 16. What's producing guinea pigs with white rough fur? 3 out of what? 16. And I'm not writing the genotypes. You can see them there in the Punnett Square. And the probability of producing smooth and white is obviously 1 out of what? 16. Are we all good? Yes or no? All right. I think what you got now is the patterns in human genetics. We talked about Mendelian genetics. Let's talk about how we make a pedigree. Pedigrees are pretty straightforward. The square is a male, and a circle is a female. Any trait that the individual has, they call them affected. An affected female is going to is going to have this filled in. It's going to be filled in. An affected female will have a blackened circle. An affected male will have a blackened in Square. It's just that simple, really. It's just a matter of either they're affected or not affected. Either that, what that means is that the trait shows up or not. Often these are diseases, that's why they say affected, but sometimes they're not. Sometimes they're just characteristics. So don't think of this as necessarily a disease. All right? So then, when you have somebody that's known that's heterozygous, and you know they're heterozygous, in other words, they are carrier of the recessive trait, what you'll see is, you'll see that half of the square or circle is filled in. Meaning one is the dominant, but it's carrying the recessive trait. Does that make sense? Yes or no, please? Someone tell me. Give me feedback. I need to know. I can't see you. Can't see your eyes. And of course, the female would be heterozygous, if it's heterozygous or if it's a carrier, we call it a carrier, right? There's the word heterozygous, by definition, means it's carrying something, right? So big A, little a, it has the little a. It doesn't show up. Little a doesn't show up if it's a dominant recessive. So we call this heterozygote. Also known as a carrier. It carries the gene, but you don't see it. It doesn't show up. The gene doesn't show up. Why? Because it's recessive. Then you have mating, or when the male and female come together. We say mating because we use pedigrees with animals as well as humans. All right. So when you're looking at a pedigree and you're talking about analyzing it, let's take a look at some pedigrees and try to analyze it. You're generally talking about first a parental generation, a P1, we call it the parental generation. You're going to see a female and a male mated together. Then this comes down here, and they're going to have children. Every shape that comes off of this is a child from this mate, from these two mating. Is this confusing to you?
Yes.
What's confusing to you about this?
Oh, no. Never mind.
You can't just tell me you're confused and then not tell me what is confusing about it. So this person, we know, has the gene, whatever it is. And we go on, and of course this female here is going to mate with someone. Here, if someone asked me, why don't you show female-female mating or male-male mating? Well, that's not going to produce an offspring, so it's irrelevant to us. Is that clear? All right. Say what? Now I'm going to ask you a question, and the answer to which is going to depend on whether you really understood your meiosis or not. So this is F1, familiar, that's a generation. So these are the children, right? F1, this is F2, F3. Do you understand that this person is not related to anyone in this pedigree? This person.
Yeah.
As far as we know, they're not really, they're married. This person also is not related to anybody in this. Now, I should go ahead and draw this in. This is directly out of your book. I'm doing an example that they used in the book. We can draw these out all day and change them any way you want, to look at different possibilities. So, looking at this, is this a dominant? Is it dominant? Is it recessive? Or is it other? What is this? And what evidence do you have that it is what you say it is?
Wait, what are you talking about? You talking about
Is this inherited in a recessive or dominant fashion? Recessive, you say. Why?
because if it was dominant, wouldn't more people have it?
No.
What?
Dominant and recessive has nothing to do with how many people have it.
I mean, it would. It shows.
What is it shows up, right? So you're actually right. It is recessive, but it's not because a few people have it or more people have it. Here's the point. This person has it, right? Where did they get the? That means this person got it from someone, the gene from somebody. Who'd they get it from? Yeah, their parents. That's what it is. See, if you say you don't know, you don't understand the concept of genes at all. You deserve not to pass the class then, right? Because if you don't understand what genes are, what are we doing here? So it got its genes from its parents. Correct?
Yeah.
So it has the gene. We know it has the gene because it has the trait. It's black or it's tall or it's short or whatever, right? It has whatever it is.
So don't go through characters and get everybody.
Correct, that's it. You got it. Because look, how can this have it? How can this child have it and its parents not have it? It has to be recessive. It's the only way. The only way that two parents can't have it and a child have it is it has to be recessive. We call this skipping a generation. The only way a disease or a trait could skip a generation is if it's recessive.
Right.
It's the only way.
What about? So wait. Never mind. I'm just be quiet.
Look, does anybody in this generation have it?
No.
And yet there's 2 children that have it. It's recessive.
You can't find it. Oh, never mind.
Now, is it, so automatically you know it's recessive. Let me ask another question. So that's right. So actually, because I know it's recessive, I know the genotype of almost everybody on here. I know the parents, for instance, let's say that it's little a for affected, and it's big A, little a for the dominant trait, whatever the dominant trait is. So that means the parents have to be little a, little a. How do I know they have to have? Because they have to have 2 little a's to give it to this person. Everywhere there's a little 2 little a's, that means both parents have to have a little a. If they're not affected, they have to have a big A. So this parent has to be this parent has to be this parent, in order for this child to have this, one of these 2 parents have to have a little a. Which parent has a little a? We don't know. All we could do with the other ones is say big A for sure, big A for sure, big A, big A, big A, big A, big A, and big A. We don't know what the rest of these are. These are all question marks. We don't know until they have a baby. Once they have a baby, or if the recessive trait shows up, then we know. So other than that, this is what we could tell about the genotype of each individual in this mating, in this family tree, in this pedigree. Is that clear? So you look at how much information you can tell just with the limited amount of information we had to start with. Does anybody have a question about how we did this? Because you will have to answer these questions on the exam tomorrow.
Well, you just okay.
You want to try another practice?
Please. I just had a question.
know how it say big A do? Why isn't it half shaded?
Why isn't it happening? Oh, that's a good point. Thank you for that. Now that we know it's big A, little a, we should have shaded, shouldn't we?
Yeah.
We should go around and do this. Half shade this one. Half shade this one. Let's see which other one we have to, we should half shade this one for sure. This one should be half shaded. Those are the only ones we know are half shaded, right?
Yes.
Okay, very good. Good point.
Thank you.
All right. Could you not, please? Let's take a look at another pedigree example real quick. Pay attention. You should be copying down the pedigree. That's what you should be doing instead of talking about whatever it is you're talking about.
You could do both actually.
No, you can't. No,
He said it's not such thing as multitasking.
no. That's not, okay.
You're childish. Okay.
I'm telling you, don't do it. I'm not gonna have a philosophical or a scientific discussion, debate here in the middle class.
I don't know what half the words mean.
So what kind of inheritance is this? It's what? It's connected to gender. Very good. Which gender gets it more often?
The males.
The males. And which kind of inheritance, what inheritance pattern has mostly males getting it? Sex-linked, and it's recessive. How do I know it's recessive? Because it skips a generation. As soon as you see it skip a generation, in other words, the parents don't have it. If it was X-linked dominant, mom would have it. As soon as you see this, pay attention really quickly.
So is Y-linked dominant?
No, if you see this, and you see it skip the generation down here, then this. If you see this, if you don't see a skip a generation, then you have to say it's sex-linked dominant. But if you skip a generation, then if it's skipping a generation, then you can say it's recessive. If it skips just one generation, just one, it's recessive. It could skip multiple generations. That's a good question. It could skip multiple generations and still be recessive. Anytime, if it just skips one or more, how's that? Then it's recessive. But if more males have it, and some females can have it, but if more males have it than females, then it's X-linked. Yeah.
Okay, so I know how like when they get married or when they die, they cross them out, and then like when they get a divorce, they cross out the little thing. So let's just say somebody got a divorce, something like that, and then they find somebody else. Where did that person go?
That gets complicated, but you won't see any questions like that on a test. How's that? That should make it, because we don't have a lot of time to get into some of those weird. In general, if you see an X through it, the person's dead. Divorces won't really matter, because the only marriages, the only mating you're going to see on a pedigree chart is mating that produces children. That's the only thing that matters. If you have a female or a male that marries more than one person and that produces children with more than one person, the chart can get really complicated. You won't see any of those on any test you'll be taking. Okay? That's a good question. I can talk to you after school if you'd like about it, but not now. All right, so that seems pretty straightforward, but there's some other weird stuff that goes on. And some of the weird stuff that you should have read about is some of the more complicated inheritance patterns like incomplete dominance. So all this up here in the pedigrees, you're either dealing with dominant or recessive. And maybe you're dealing with X-linked. In other words, the gene is on the X chromosome, and we talked about that. That's what linked means. Linked means it's on that chromosome. X-linked means it's on the X chromosome. The gene is on the X chromosome. So with incomplete dominance is one of those non-Mendelian, right? This is the kind of inheritance pattern that doesn't follow the simple dominant or recessive. So what would an incomplete dominant look like? First of all, we don't use big R, little r, for instance. Let's just use R as our letter. We would use big R and then big R prime, and then we would use let's just use that. So if you have, let's say, a red flower and you combine it with a white flower, you mate these together, what you could get is you could get all kinds of combinations. One of the things you can get is a red flower, and that red flower would have an RR. If this red flower is R, if the two together, well, you can't have. See, this would never happen. So let me clarify. You would never get this. These two could never come together and form a red flower. All of them would be pink, is correct. And the reason is that red is RR, white is R prime. Okay. The prime is this little hash mark. That's what they mean by prime. So all of their children are obviously going to be R prime, which is pink. Does that make sense?
Because red and white.
Well, because you're getting one of each. So they don't hide each other. See, in a dominant recessive, the product of one gene hides the product of the other gene. In incomplete dominance, they don't hide each other, they blend. So the red and the white blend to pink. So they are all pink. Now what happens if this male then mates with another pink female? What kinds of different... You could end up with someone who has a big R and a big R, right? Because you can give two big R's, which would make it what? Red. You could end up with one that has big R prime, R prime, which would make it white. Yeah. Or you could end up with a flower that or a plant that has big R prime, which would make it pink. So you could, if you take 2 pink flowers, you can get 1 of each.
How do you know which one's prime and stuff?
I don't know what.
How do you know which one is prime? It's a common next to prime.
No, I know it's
Like,
like,
how do you
I know,
know,
but, like, how do you choose that it's prime or not?
Yeah.
I see.
Because in the chapter, it ain't really saying that. It just said, it just shows prime.
Good question.
Thank you.
You're asking, how do you know it's true breeding or not? You know, how do you know it's homozygous? So your question really is about, how do you know if it's homozygous one or homozygous the other or heterozygous? So pink is heterozygous. We know that because if we take 2 pink flowers together, we can get any one of the colors. That's how I know that these are. Now, how do you know which one you call R and which one you call R prime? It doesn't matter. You choose which you call R prime, which you call R. What one is going to be the white, if I mix white with white, I'm always going to get white. If I mix red with red, I always get red. But if I mix 2 pinks, I can get any 3 of the colors.
So red is recessive?
Red, not recessive. Red is pure, what we call true breeding. In other words, it's homozygous. White is homozygous. Pink is heterozygous. What's cool about this is that with incomplete dominance, there's no hiding. There's no carrier. If you have the gene, it shows up. So if you have the 2 genes together, you see pink, you know it's heterozygous. If it's homozygous R prime, then you know it's white. It's white, so you know it's homozygous R prime, right? And if it's red, you know it's homozygous R. So it's really easy. So if somebody ever asks you in a like, let's say on tomorrow's test, the genotype for an incomplete dominance phenotype, then it's easy, right? As long as you understand what the outcomes are from the mating. All right, so that's not the only other kind of weird inheritance we have seen. There's incomplete, but there's also what we call co-dominance, right? And then there's multiple alleles. So with co-dominance, you would have, again, that big R prime, big R prime. Let's say this is red again, and this is white. And the thing is that when these 2 come together and they produce a big R prime, they don't make pink, they make red with white dots. Or let's call them spots. So both of them show up, they both show up independently now. They don't mix the way incomplete dominance does. This is called co-dominance. In other words, they both show up together. Blood type has a little bit of this mixed in. Blood type's a little confusing, so we're going to look at blood type in just a minute. Blood type is actually a combination of multiple alleles and co-dominance. Did you say it's not?
No, I was talking.
Oh, you're talking about something else. Okay. So you have these genes on chromosomes. Remember, you have these chromosomes. They have these genes. If you have the A gene, what blood type are you? A. If you have the B gene, what type are you? B. And if you have the O gene, what type are you? O. Ah, there is a B because you have 2 copies of each chromosome, so there's 2 copies of each gene. So if mom gave you A, and dad gave you A, then what did you get? What's your blood type? A. But if mom gave you if mom gave you A and dad gave you B, what blood type are you? AB. If mom, is it time to go?
Yes.
All right, so here's what I'm gonna do for you.
Talking about ourselves, we really have a minute.
Okay.
Somebody to teach.
Are you guys gonna put stuff away without me telling you?
Okay,
Listen to what he
this
said,
front
bro.
table.
I just need y'all to listen to me without putting stuff away inappropriately. That's all. That's all I'm asking. But I'll wait for you. I'm not gonna be rude.
Y'all keep
Just
on.
say if they don't hear it, that's their fault.
Right, really.
Except for the consequences.
So, seeing as some of you don't feel comfortable with all the content, I'm gonna do you a favor, and I'm gonna move the test to Friday. I'm gonna tell you that I'm concerned about doing this, mainly because some of you are not getting your work done. I can see a lot of you've done your reading, good job, and you turned in your work, I saw it. But you gotta understand the material. So when I post this tonight, please get the help you need. You have tutors here twice this week. Take advantage of that. Now, if one parent gave you an O and the other parent gave you a B, what do you have? You have B. The O is what we call recessive. So you do have B blood type, that's the phenotype. This is the phenotype here, AB. This is the phenotype B, but you do have the O allele. Do you see it? O, for the record, the way blood type works is you have blood cells, and if you have an A blood type, you have this thing that might look like this on the cell surface. If you have a B blood, that's A. If you have a B blood type, you might have a protein that might look like this on the cell surface, okay? And there'll be a bunch of them. If you have an O blood type, you have nothing on the surface. There is no surface marker, no cell surface protein that your immune system will react to. So O blood type, O is recessive, because if you get an O allele, there's nothing, the O is not going to produce anything, but the B will. So effectively, you are what blood type? B. Whether you're BB or BO, it doesn't matter. Are we clear? All right, so there's 3 blood types, A, B, O. A and B are dominant over O because O is no cell surface protein. O is there is nothing there. Where B, you'd have a B, and A, you'd have an A, so they're co-dominant with each other. These two are co-dominant. And O is recessive to both A and B. So if you have an O blood type, what are your genes? If you have O blood type, what are your genes? They're recessive, so what are they? You have OO, is correct. You're homozygous recessive. If you are O blood type, then one gene has an O. You got one O from mom, and you got one O from dad. Yeah, you are OO. Does everybody get that? Is anybody confused about that at all? Now the notation, the way we talk about notating it, when you're talking about these cell surface proteins, we call them I. The letter that we use to designate them is I. So the carrier, the A blood type would be I, and then if you have the A, it would be IA. If you have AA, it would be IA. What's another way to get A blood type? You can have IA, right? You have, those are two ways, IA, and then IA or what? Right, or O. Now, instead of saying IO, what we say is we use the I again, but it's little I, because it's recessive. Are we clear? So really, not little I, it would just be little I. Little I because it's recessive. Does that make sense? Yes? Okay. So B could be, you could have an AB, IB, or you could have a what? B, little I. AB has to be A what? AB has to be what? It has to have a B. It's the only way you're going to get AB. There's only one genotype for AB. Everybody see that? Yes or no? Please answer. Let me know. I can't see you if you're nodding. Yes. Remember, I got lights in my face. I can't see you. So you have to respond. All right. So how do I get, so this one gives me A blood type, this one gives me B blood type, this gives me AB blood type. How do I get O? Little I, little I. Is that clear to everybody? So that's why we call this codominant. That's why they're codominant, because if you have the A, it shows up. You have the B, it shows up. So A and B are both showing up. We call them AB. However, little I is recessive to both A and B. All right. So let's talk about polygenic inheritance. Skin color, for instance, is one example of polygenic inheritance. What do I mean by polygenic inheritance? Anybody do the reading that might know what polygenic inheritance is? Inheriting a trait that depends on more than one gene. Not allele. When we just looked at with ABO blood type, we're talking about 3 alleles, right? One gene with 3 different versions of it. So there's a really clear distribution there. But what happens when you got many genes, for instance with skin color? So let's compare that to something else that's not polygenic. Let's look at something like sickle cell. Right? Sickle cell is what? It's either a dominant, this or this is going to get you what? No sickle cell. Is that correct? Mm-hmm. And then little s is going to get you what? Sickle cell. So there's only 2 options, 1 or 2. That's it. With multiple alleles, you might have as many as 3 options as far as phenotypes are concerned, correct? When we look at codominant and incomplete dominance, again, there's 3 options. By the way, when there's multiple alleles, you can have many different options depending on how many different alleles you have. Does that make sense? But With polygenic inheritance, you have a whole range of options. So we talk about something like skin color. We have light to dark. What does dark mean? What does light mean? Who? What? You can get a. We could pull out cards and we can look at. We have something called a spectrometer. We can measure the shade of any color. You could actually. There's some paint stores now that can do that. Tell you exactly what color you have, etc. We don't need to do that. We just can we all agree that there's light and then there's dark? Yes. All right. So it turns out that if we had just one gene with multiple alleles, we would expect to see something that might look like, something that might look like this, right? We'd have either dark or light, and if there's 3 alleles, there might be dark or light, and maybe a third option. But we don't see that with human skin color, do we? What do we see with human skin color? This is not what we see with human skin color. Does anybody pay attention? Have you seen humans? Have you looked at their skin color at all? So what do you see?
It's one color.
Do we see one color?
Well, not always.
Never. Look in this room at each other. We can turn the lights on, and nobody in this room is going to tell me that you're all the same color.
I thought you meant like on a person's body.
Oh, no. I meant among people. It's a good point. Among different individuals. Look in this room. Is everybody here the same color? Light or dark?
We all got on blue?
No. What you see is this distribution. See something like this. You see something that has some really light people, some really dark people, and then everything in between. That's because there are multiple alleles. Multiple genes. There's more than one gene. In fact, with skin color, I think there's something now around 6 or 7 genes involved in skin color. So you cannot talk about dominant or recessive or even codominance or even incomplete dominance the way you see in flowers. No, with human skin color, there's multiple alleles dealing with the shade of skin tone that you are. So there is no one skin tone. Is that clear? There's no black or white or black, white, middle, like some people will give you all kinds of different colors and shades and what have you. That just doesn't exist. There's actually 64 shades that have been identified in human skin color. 64 shades of human. So you can't really say black or white. You have to say, there's 64 races then? So when you use race and you use skin color as an identifier of race, that kind of, it defeats the purpose. It doesn't make sense. The book does use the term white and a Caucasian, and I think, right? The chapter reading does use the word Caucasian, doesn't it? And I think it uses, I think it might actually use the word black as well. That's not an appropriate term to use when you're talking about genetics, whether it's skin color or not. Well, you could, but even African American, there are plenty of people. In fact, one of the greatest African American writers, one of the greatest African American writers of your time, of this time right now. Oh, what is his name? So the author's name's Malcolm Gladwell. He's a writer. He's born in 1963, he's from my generation. But when people say, you say African American and skin tone, that's his skin tone. Does that fall within your paradigm? Does that fall within your view of African American, that skin tone? Okay, but you're not, Okay, you're not being very scientific. So what I'm asking you to do is really break it down when we're talking about skin tone. Skin tone is not appropriate when discussing race. Race doesn't exist. There's no set of qualifiers. There's no objective set of identifiers like you have with blood type, like you have with flower color, where you could say, okay, this is an African American, this is a Caucasian, this is an, whatever. It doesn't exist. So the appropriate term, there are cultures, African American culture is real, and there's a population of people, you could say the African American population. But to call it a race, define a race by a certain set of physical characteristics, when those physical characteristics there are many, many different numbers or individuals in that population that don't have those characteristics, is disingenuous, right? It's confusing, isn't it? So really, when you're talking about race itself, it's not a biological term. It's a social term that governments use to classify individuals, but they're not useful in science. Caucasian is not a real thing. It doesn't really exist. Hispanic is not a real thing. It's a definition that's used by governments to classify groups of people that are very genetically diverse. African Americans are genetically diverse. Culturally, it's one culture, but there's a genetic diversity within African American culture. The population of African Americans in the United States is genetically diverse, so you can't really point to characteristics, to phenotypes that one could put and identify something that you might say is a race. The same with Caucasians. Caucasians in this country, in the world, are very diverse. You have light skin, like Caucasians from where? Sweden, Germany, British Isles, Ireland, where? Italy, Canada, United States, Spain. There's Caucasians from all over the place, and what people think of Caucasians, they think of light skin. That doesn't work. The populations are different. You cannot tell me that genetically the population of Spanish individuals is similar to the people from Switzerland, right? If you looked at those two people, they both could be white, they could both have straight hair, they could both have blue eyes. But one would have Moorish genes in them, the other one wouldn't. Do you know who the Moors were? The Moors were people from northern Africa. Spain was ruled by the Moors for 500 years. You can go to Spain and see Arab influences in the architecture. You can see, and you look at the genes of the Spanish in Spain, a European country, and you will see the influence of the Moors in that population. You will see less of that in Switzerland. In Ireland, you'll see a different mix of genes. And yet, we would group all those people together in one group. Look at Russians, and you'll notice that their genes are influenced by the Asian population, the Asian of up in Mongolia. The Khans ruled most of Eastern Russia for a very long time. Do you know who Genghis Khan was? You've heard of him? Yeah. He's Asian, and his people ruled Eastern Russia. They moved all the way to Rome. So when we're talking about, and by the way, bless you, when you're talking about genes, this conversation gets, the identification of race becomes useless. So scientifically, race is not a thing. populations are. Populations are. Who, where, what baby had what baby, and what genes gene mixture gets you what phenotype. So when we talk about skin color, we know there's at least 8 genes, probably 64 different, and who knows how many alleles. I say 8 genes. How many different alleles? I have no idea. So now we got 64 different shades, at least minimum. Question or comment? It's what? I can't hear.
Like Chinese people.
Yeah, Asian people, China, Japan, Korea, they're 3 very different populations. They have been separate for thousands of years. I can promise you the genes in each of the 3 populations are very different. They would not mix. Probably you didn't get a lot of intermixing between a Japanese and Chinese family, for instance, historically. And Koreans are right on the border between China and Korea. They're very, very separated culturally. So it's highly unlikely that you get a lot of intermixing between Koreans and Chinese or Japanese for a very long time. For China's been around for 1,000 years. So to say that they're all Asians, put them all in one group called Asians, is ridiculous. In fact, Asians include, get this, are you ready? Chinese, Japanese, Korean, Indian. Indians are Asians. Filipinos. How do you do that?
I didn't know Indians was Asian.
Yeah, they call them that, but it doesn't. It's only understand. Are you guys listening to me? Are you listening to me? The word doesn't mean anything. The word Asian is only used by governments to classify groups of people because of geopolitical standing or where they are in the world, and that's it. It doesn't have any real connection to biology. Is that clear? So you need to understand that when you talk about race, biologically it doesn't exist. When you hear racists talk about the purity of a race, that's ridiculous. That's just not, it's not true. All right. So that takes us through the content so far. I believe that catches us up. Does anybody have any questions on any of this? Because what I have then is that the next thing we're going to talk about is what your test looks like. You should look at the end of your chapter assessment. It has a number of vocabulary reviews. There's a vocabulary review section, understanding key concepts, constructive response, thinking critically, as multiple choice problems. So there really shouldn't be any question what kind of questions are coming on the test. You've had practice problems, and you're at the back. You did not have to turn those in. Why is everyone putting stuff away? It's 3:06. I do it every day. Every day I get to say, every period it's the same thing. Can I be the one that dismisses you? Is that okay? Okay, so let's be clear on a couple things. If Bell does not dismiss you, I dismiss you. So let me be clear on that. I can't be any clearer.
Okay, you just messaged. I'm not even trying to play
Like, I'm not, it's not about being boss. Somebody has to be responsible for you, and that's me. Whether I like it or not, whether you like it or not. So I'd like you to sit down. I'd like you not to put your stuff away until we're done. I can't tell you how many of your parents are going to say, well, what's on the test? My kid didn't, listen, when you don't do well on an exam, I can tell you what you say and what your parents say. I didn't know that was on the test. How was you didn't give me those questions. That's not fair. You didn't explain the answer. These are the words that you and your parents use when they come to see me. And I'm going to tell you right now what my answer is going to be to you and your parents. I've told you what's going to be on this test. I've gone over all the practice problems that I have assigned to you. I went over the chapter now. I'm telling you there are practice problems at the end of the chapter. If you want to practice them or not, they are there. I will be here tomorrow morning and during lunch if you need grade conferences. If you need them, I'm willing to work with whatever you need to work on. There are tutors here after school in the dining hall from the medical school. Here's the problem with going to tutoring at the last minute. If you show up at my door and you're number 5 and we run out of time, that's all there is. So when you guys wait till the last minute to get tutoring or to get help or do whatever it is you want to do, if you don't get the service, you don't get the service. There's only so much time and there's limited resources. So, does anybody have any questions about polygenic inheritance? Let's be clear, just on polygenic inheritance before we're done. When you see here light to dark, you have people that are mainly, most people are in the middle. You have light-skinned people, darker-skinned people. But because you have this distribution, we call this a distribution, we call this a polygenic trait. Now, what about this? What about height? How tall or short? Please leave my room. Close the door. Wow. Close my door. Thank you. How's that girl gonna walk in this room? That's right. Now I'm gonna write her up. Okay, so now that interruption is over, let's be clear. Height is the same way. You have the shortest among us would be here. You're not the shortest, and you have, if we put a bunch of different populations and we looked at the height differences among them, and we looked at all our people from all different places, what we would see is we'd have the tallest on one end and the shortest on the other, and we would have the same kind of distribution. The reason we have this distribution is there's more than one gene. This is called multigenic inheritance.
Are there specific kind of genes? Like
There are multiple genes that are involved in height. So just like with skin tone, you could have there is a recessive, just like with blood type. You have blood type, remember you have A and B, they're codominant, but O is recessive. Well, just like that, most skin color among humans is dependent on a combination of many different genes, but you do have this one skin tone, one color that we call albino that is recessive. And just like albinoism, you could have dwarfism.
Okay, I'm not a dwarf.
All right. Dwarfism is more than just a shorter person. There's other phenotypic, there's other consequences, just like with albinoism. But dwarfism can be dominant, and it can be recessive. But the point is that it's not part of the spectrum as far as height, because the genes for dwarfism can take over. You might have a multiple combination of genes that might normally make you tall, but if you have a specific form of a gene, whether it be dominant or recessive, it could make you a dwarf. So genetically, these things are complicated. But when we're talking about shortest to tallest, when you have this kind of gradation, like you do with skin tone, like you do with height, we call it multigenic inheritance. And that's it. That's all that's going to be on the test. I'll be posting this, and we'll move on.